So what's kind of interesting is that there are strains of fungi that are actually parasites of the rust parasite. So we call them hyperparasites because they're parasites of parasites. And so what they
just
do...
turtles all the way down. Right?
Yeah.
I can't think of any more human activity than conducting science experiments.
The game I play is a very interesting one. It's imagination in a tight straitjacket.
The beauty of a living thing is not the atoms that go into it, but the way those atoms are put together.
What I always think should be the basis of education is not answers, but questions. We should teach kids how to question.
So my name's Michelle Moffitt. I'm a senior lecturer at Western Sydney in microbiology. So I guess my current research is in looking at fungi. So we're interested in small molecules that these fungi produce and what their effect is on other microbes. So we're looking for compounds that may be antifungal, so they might kill other fungi, or antibacterial, so they might kill bacteria. So that's what we look at the moment. But it's kind of a deviation of where I've come from, really.
Right.
Oh, that's interesting.
Okay, so just going back in time, walk us through, or maybe you can...
So you said it's deviation from where you came from.
Yeah,
so the next logical question is, where did you come from?
So I'm not actually a mycologist. I never really did fungi before, never researched fungi before. So originally I've always sort of worked on bacteria, and more specifically I've worked on cyanobacteria, which are photosynthetic bacteria. Right. People call them blue-green algae sometimes, you might hear them called that, because, you know, if you look at them under a microscope, they look like algae, but they're not actually eukaryotic, they're bacteria. So I was looking at toxins that are made by cyanobacteria when they bloom. And so that's where I started my research journey, is looking at cyanobacteria.
That's cool. Let's maybe even go back further than that. Were you always interested in science?
Yeah.
How did that happen?
So basically, I have a pretty good memory of this. When I went to high school, you know, year 7, first science lab, we go and sit in the lab, and there's a sink, and there's a tap and a Bunsen burner, and we boiled water, and I was like, oh, this is cool. This is what I want to do for the rest of my life. Work in a lab, boiling water over a Bunsen burner.
Oh, that's cool.
That's interesting, because when I think I've always been interested in science, not even knowing it, but when I first went into high school, year 7, I walked into my chemistry. There was just a science lab, I suppose. And I remember that night going home and having a dream of me being in a lab coat in that exact same science class. I remember I was building A2, and I was just mixing chemicals. I'm like, oh, this is the coolest thing ever. This is what I want to do for the rest of my life.
Yeah, totally.
Yeah.
Yeah, I could never understand that people found that boring. Like,
Yeah.
Well, I'll tell you how. No, I went into high school really loving science and kind of been really interested in how the world works, but I kind of got turned off.
Oh, really?
Yeah, in high school. There was a lot of just fact learning for me, and it just wasn't interesting. Possibly the way it was taught, but
yeah, I think teachers are a big thing. So I had some pretty amazing teachers in high school. A really good chemistry teacher, a great physics teacher, and they really had a passion for what they were doing, and I think that rubbed off on me as well.
Yeah.
I think if it was boring, I probably would have lost that passion.
For me, there was too much of, here's the textbook, and you have to learn all these facts and be able to reproduce them in the exam, and there wasn't enough like science as a method, and we use it. Like we did the whole, I remember just having to like write out methods by rote off the board, and then go and do the experiment now, and then just write up results. And there was pages and pages of writing just in every science lesson. And I'm like, what am I doing? I'm just copying stuff off a board. That was my high school science. And I didn't do science for the HSC because of that. And so it really
that's
changes.
a shame.
Yeah.
One of my high school teachers had actually done honours after her degree. I think, that's something that is really positive for high school teachers. I think if they can kind of have a bit of experience in research themselves, I think that would, get them more enthusiastic for their subject and pass that on to their students as well.
I suppose it gives them a better understanding of science and how it's actually done versus going through an undergrad in science, for instance, and just, these are some facts, these are some biochemistry, here's some microbiology. You don't really get an appreciation of, how did these people actually find out these facts? What were the approaches? What was the process like? I think that's really important, those stories, and it gives context to the facts themselves and makes it more interesting. Right. So you were interested in science, but how did you get involved in microbiology, and what took you in, brought you to microbiology?
Well, that's
obviously not the smell, right?
No. Actually, it's funny you say that, because that just brings a memory to my mind that I hadn't thought about for a long time. But when I was in high school, I did that you do work experience, and I did work experience in a lab, and this woman was testing the sterility of the work environment or whatever, and every morning she would make the agar, and it just smelled so bad.
Yeah.
But actually I've forgotten about it. So it didn't turn me off. But then when I was finishing high school, I kind of wanted to do research into pharmaceuticals. I wanted to discover new drugs and things like that. And I kind of thought, well, maybe I should do pharmacy as a degree. And I talked to some pharmacists, at the Uni Open Day, and some science people, and the science people said, don't do pharmacy, you'll find it really boring. And so I went and worked in a pharmacy, and I was like, pharmacy is not for me. So I just did a straight science, advanced science degree. And I went into pharmacology, so that was my major. But all the pracs that we did in undergrad were on animals, and I didn't like that.
Right.
And I did micro as kind of a, sub-major.
Right.
And I realised, no one cares if you autoclave a bunch of bacteria and they die. So micro was more my speed.
Yeah, well, I have a few friends that do work on rats, and they have to euthanize them. Yeah. And it's just really depressing. They're like, this is the most depressing stuff. Like, you have to go and kill these rats or mice or whatever. Yeah. And so there's definitely an ethical load that you have to carry when you're doing that type of work. Whereas with microbes, even cells, we work with cells. And we're like, okay, we just abuse them.
Take that.
Microbiology was basically a way for you to do research, and but not necessarily do work on animals.
Yeah.
I shouldn't say research.
On fungi, you mean?
Yeah.
Not do work, sorry. Go on.
Not work on actually you just said animals. Alex needs to pay attention
to
what...
I thought you were leading to fungi.
No, not yet. Because you went from an advanced science degree into, I'm assuming, an honours?
Yeah.
Okay, and why did you do that? Is it because you were interested in actually... So how did that how did that fit with your initial idea of wanting to develop drugs?
I guess at that stage, at the end of third year, I sort of... I knew I wanted to do research. I just wanted to work in a lab. That was really my goal in life, was I wanted to work in a lab. So I wanted to do honours. I guess it was part of the advanced science degree anyway, was you just did honours at the end. And I kind of scoped out a few academics at the uni where I was at, and I ended up just in a lab that was working on these cyanobacteria. So actually, when I was doing my undergrad, I wasn't an ace student. I was so for anybody out there who's doing their degree and not getting straight HDs, that's okay.
Yeah.
You don't have to be a HD student to be a good scientist, I think. But I wasn't the best student, so I wasn't taken by the best professors, and so I ended up in a lab with a young guy. He was just starting out his career, basically. He was only just out of finishing his PhD, not that long before I started. And so he was working on cyanobacteria, so that's what I did. And so, yeah.
So tell us about what were you researching?
So cyanobacteria bloom in water. So they basically, whenever there's a water system that has increased levels of things like phosphates and nitrates, then that triggers these cyanobacteria to just overproduce. And so they can produce, some of them produce toxins when they bloom. And so the one that I was working on was a strain that it blooms in the Baltic Sea. So if you take a picture of the Baltic Sea from space in the summertime, you'll see the bloom from space. It's huge.
Yeah.
But it also blooms in South Australia, where they sometimes get their water supply from estuaries, lagoons and things like that. And so if you have cattle that are drinking from that water, they can get sick.
Right.
Basically, the toxin affects the liver. And they say that people in, some of these third world countries actually get higher incidences of liver cancer because they're constantly exposed to low levels of these toxins.
So it's
Correct me if I'm wrong, but was it cyanobacteria that were responsible for one of the first major extinctions on the planet? Is that right? From producing the oxygen? Because they were the first oxygen producers, weren't they?
Yeah, they were the first oxygen producers.
And when they first produced oxygen, it was a poison for most of the organisms that were on the planet, and resulted in mass-scale extinction.
Yeah, but at the same time...
these little buggers are...
But if it wasn't for them, we wouldn't be here.
True.
So they were really the trigger for eukaryotic.
Expand on that a bit, because I was going to probably ask that question soon.
Yeah. So... I guess cyanobacteria evolved, so they're photosynthetic, and so a byproduct of their photosynthesis is oxygen. And so over time, the oxygen accumulated in the atmosphere, and then that triggered, I guess, the evolution of aerobic growth.
And wasn't it also cyanobacteria that gave us plants, in a sense? Because
Yeah, so the chloroplasts that are inside plant cells are basically cyanobacteria. So the plant cell engulfed a cyanobacteria, and then they had a
means of producing energy through photosynthesis.
Yeah.
That's really interesting. So in your research, you were at that time you were looking at what exactly? So you were dealing with the cyanobacteria.
So we were looking at the genetic basis for how the toxins are made. So at that time, there wasn't the mass genome sequencing that you see today. So we were sort of picking out bits of the genome and trying to figure out how those toxins were made. So, so that was what I was doing my PhD on.
And was that your PhD as well? So you went...
Yeah, I started off doing it in honours.
Right.
And in honours, I was just kind of scoping out the species and looking at how different strains that are collected from across the world, how they're related. So are the Baltic Sea strains related to the Australian strains? So it turns out that they're all kind of a global, there's no difference really between all of these strains that we see blooming across the world.
Oh, that's interesting.
Yeah.
So that was my honours.
Right.
And then I went on in my PhD to look at the genes.
Specific
that make the
genes
toxins.
that make
Yeah.
the toxins. And so did you isolate the genes that were involved?
Yeah. So I sequenced the genes. So the nice thing about these genes that they cluster together on the genome. So once you find a little piece of it, you can kind of sequence around that and then find the entire gene cluster is what they call it.
Right. And so
Yeah.
why was it important to find the genes for this particular toxin? Or what utility did that knowledge have?
Well, I guess it's the first stage of then trying to understand why these toxins are being produced. So if you're in the water industry and you want to know when the bloom is likely to occur, when is it going to be more toxic than other times, then it's important to know what triggers that production in the cell. So if we can find the genes, then we can look at things like what turns the genes on and off. And then you can sort of inform the water industry and say, okay, when this, whatever is high, then that's going to increase your chances of a toxic bloom.
Yeah, interesting.
Okay.
So how do we?
And I guess the other thing is that it's good for the water industry to then have that as a tool for genetic testing. So they take a sample of water.
Yeah.
Is this water toxic or not? I can do a quick PCR test, and that tells me, okay, that bloom's toxic. Okay, we need to ramp up purification of the water so it doesn't go into the water supply.
Yeah.
That's interesting.
So then how did you transition from cyanobacteria into fungi?
Can you get a little bit closer?
Yeah,
to the mic?
sorry.
So,
Yeah.
Yep.
So how did I go from there? So I was in the States for 3 years, and then I was working on other bacteria that produce other molecules. And then I came back to Sydney, got the job here, and I was still working on cyanobacteria for a while. But the downside of cyanobacteria is that they grow pretty slowly. So they're difficult if you're working in a small lab. Just so happened that a friend of mine contacted me about this project that he wanted to get off the ground and said, would you be interested in working collaboratively on it? So so I said, okay, let's do it. So that's how we started off on the fungi. But it's related because the compounds that we're looking for in the fungi are made in a similar way to the toxins, how the toxins are made in the cyanobacteria. So they're very similar in their system.
Yeah. Can you unpack that a little bit? So
Yeah.
how are they similar?
Okay, so basically they're all small molecules. And they're all produced as what we call secondary metabolites. So they're not compounds that are essential for the growth of the microbe. They're produced when the microbe is flourishing, and they don't require their primary growth anymore. So yes, as I said, they're small molecules, and as I mentioned previously, their genes are clustered on the genome, and they're made not via the ribosome. So the ribosome would normally make proteins and peptides. So these are often peptides or sort of fatty acid type molecules, and they're made on these huge enzyme systems. So these enzymes, they'll select an amino acid and incorporate it into a small molecule. And then there are other enzymes that then modify these small molecules to make them active against their target, whatever that target is.
Okay, that's interesting. I suppose while we're on the topic of fungi, we might have some audience members that don't know much about fungi. Can you tell us what it is? And let's start there before we transition.
Yeah, what is a fungi?
What is
Most
a
people
fungi?
probably know it's a mushroom, but that's like where it stops.
So fungi are eukaryotic microbes, so they're very similar in their cell structure to us. Okay, so they can be unicellular, they can be filamentous, and then sometimes they can grow into things like mushrooms or truffles. So, yeah, that's...
So most people think that, fungi are plants. Right? That's not really the case.
No. So traditionally, I guess they thought that fungi were related to plants, and fungi are kind of in between the plant and the animal kingdom. So, they're sort of in between. They're decomposers naturally in their environment. They're usually decomposing dead material. So that's one of their big ecological kind of benefits in an ecosystem.
Because that's then becomes food for plants and things like
that,
isn't
exactly.
it?
So in the soil, they'll break down the dead plant material that's around, and then that all has to happen outside the cell of the fungus, because it sort of ejects these enzymes to break down the material.
And humans technically do that as well, don't they? Yeah, we release enzymes in our stomach, intestine that is technically outside our body.
That's right. So, and then, they take up whatever they need, and then whatever's left over can go into the soil and feed bacteria, can feed plants.
So plants actually eating the leftovers of...
Yeah, pretty much. Yeah.
Oh, that is interesting. I didn't know that. I thought... Okay, well, that's fascinating. Well, I was listening to a podcast, and they were talking about... so besides just decomposing dead matter, they're also... they're connected to trees, to the root systems. And they not only exchange nutrients, so I think it's nitrogen for glucose or sugars, right?
Yeah.
But they are also able to connect to different trees at the same time and actually modulate what trees get what nutrients.
Yeah, it's pretty crazy stuff, huh? So there's like this whole signaling, they can talk to each other through these molecules that signal, to the tree or to other fungi, and then they can influence their growth. And then, so what's interesting as well is that I guess it's possible that some of these small molecules that the fungi produce could actually be taken up by the tree and then used by the tree as a defense mechanism as well.
Oh, okay.
By the plant, I should say.
So it's almost like, in a sense, the immune system of the plants.
Yeah. It's one way of protecting itself.
Is that like similar to how microorganisms are in a way connected to our immune system as well, in that our like particular microbiota, the microorganisms we have in our gut and things, maintain a particular environment which is hostile to pathogenic microorganisms.
Yep, definitely. Yeah, and that's one thing about microbiology that since I've been teaching it here, I've really seen that we've discovered so much in the microbiology world that wasn't known when I started teaching here. And that's amazing to me that this kind of field is really taking off, and we're starting to see things like this, that how important they are in a human's immune system, in a plant's biology, in an ecosystem. It's
almost like we're just the machines that the microbes are using to carry them around. Yeah.
Exactly.
I think it's even, I don't know the exact numbers, but I think the number of microbiological cells that we have in our bodies is the same as the number of cells that actually have our genome.
Yeah. So they say that...
They weigh less because the cells are actually smaller, but in terms of sheer numbers...
It's something like a couple of, maybe a kilo or of our body is microbes, or maybe more. I can't remember the numbers either, but it's pretty crazy.
I think the ratio is like 10 or 100 to 1.
I think that's what they thought it used to be.
Is it more?
But no. I think it's actually there.
Yeah.
They worked out it's actually pretty equal.
Yeah, there was an original estimate, and then now the scientists are saying, oh, maybe that's kind of a bit over the top. But
Yeah.
I think
even
if
the same is quite amazing.
yeah.
50% of you isn't you.
Yeah.
Yeah.
Yeah, it's pretty amazing.
That's crazy. Well, we had Dr. Alison Moore, and she does some work with, I was going to say feces, but
Well, yeah.
yeah, a little bit. Yeah, and it's just amazing how important just gut health is, that brain to gut to brain connection, and that some people who have issues actually get fecal transplants.
Yeah, that's a really big thing at the moment. It's pretty amazing. So, the bacteria in your gut can help to send signals to your brain. It can, so if your gut microbiota is not right, then you can try to fix it by transplanting different microbes in there. Yeah.
I wonder if you'd know this, but is it that the bacteria are producing certain neurotransmitters, or they are stimulating the production of neurotransmitters by working with the nerve cells that are around the gut system?
So what he's asking is this: so if the neurotransmitters are coming from the bacteria, or if they're just stimulating like the neurons in that system.
I'm not 100% sure on this, but my understanding is that they produce molecules that are the same structure as neurotransmitters.
Okay, yeah. Interesting. Yeah, we were thinking that the other day. But maybe we can move on a little bit, because we talked about the good and bad of cyanobacteria, and we've talked about the good of fungi, but there's also quite a lot of pathogenic fungi, isn't there?
Yeah.
For humans and in agriculture.
So that's where this project really stems from, is that, a lot of the infections that we get as humans are bacterial, but if you have something going on with your body, if your immune system is low for whatever reason, then you're more prone to get fungal infections. But in agriculture, plants are very prone to fungal diseases. So we see a lot of fungal diseases in agriculture, and they can be a problem.
Sorry, a little bit of a tangent. I'm just quizzing you a lot today, but but by any chance, would plants be more prone to fungal infection because they have that close relationship with fungi? So their systems are designed to have a close relationship with fungi, so that leaves a niche for other fungi to be pathogenic.
Yeah, I think so. Yeah. I think that's probably it. And they're in an environment where they're, they're interacting with them more closely. Yeah, whereas our immune systems are pretty good at keeping them at bay. And
Okay, so let's transit. Just speaking about pathogenic fungi. I've had, this is probably gross to say, but I've had...
Hang on. I'll get the beep button ready.
So I do Brazilian Jiu-Jitsu, and that's skin-on-skin contact. It's wrestling, grappling.
Yeah.
And I've had ringworm. I've caught it from people.
Yeah.
And that is a pain to get rid of.
Yeah.
Can you talk about some of the difficulties of treating things like fungi, pathogenic fungi, in relation to human beings?
So we talked before about how fungi are kind of sort of very similar to plants, and also similar to animals in their basic biology. And so when you treat a bacterial infection, you are targeting a part of the bacteria that is very very different from the human cell. So bacteria and human biology is very different. So you can make drug targets that are very specific to bacteria.
But they have no impact on humans.
But they don't hurt humans because there's no similar system in the human cell. So with fungi, their biology is very similar to our biology. So if you take a drug that, might target a pathway in the fungi, there's a very good chance that you have a similar pathway in the human cell. And so you can find fungal specific drugs, but they have a higher chance of being toxic to the human cells. So Yeah, so they're often very difficult to treat.
They're definitely a pain. Is that also the case because you said earlier that fungi are, in a midway point between plants and animals? Is that also the case with plants as well, where if you try to treat them more often than not, you'll actually, because there are analogous systems in the plant cells, you'll have a toxic effect on the plants?
Yeah, I think so. I guess my understanding of that side of things is not as good. But yeah, I guess we're a bit more sensitive though to, to a toxic side effect. Whereas, you might put a fungicide on a plant and it might make the plant a little bit sick, but it's not as bad as it getting the disease.
Because they are, I think fungi are more closely related to animals than plants, aren't they?
Yeah. So, there's a possibility we would have that overlap, but like I said, we're more sensitive. We don't want to give ourselves a drug that's going to make us sick, but we don't,
Right. And also, if like a branch of a plant dies, it can grow another one.
Yeah.
If your arm falls off, see you later, buddy.
That's right. That's so true.
There's no replacing that.
No one cries when the plant loses a leaf.
No. Everyone cries when they lose a pinky. What are some typical targets for fungi? Would you be able to talk about that?
Because I know in bacteria, I think it's the cell wall and things like that.
In bacteria, it's things like the cell wall, whereas in fungi, I guess, they have their own cell wall, which is different to ours, which is chitin, so that could be a possible target, but there are other targets that target things like cholesterol synthesis, germination is another one. So there are some targets that we can look at.
Right. Let's talk about rust and your current research. Can you give us, we heard you talk about it during the research development showcase, and that was really interesting. Give us a background on rust and how your research fits into that, please.
Okay, so rust is a fungal pathogen, so it's an obligate parasite on plants. And
what's an obligate parasite?
It means that it can only grow on a plant.
Yeah, so the only way it survives is by
being a parasite
on living off plants.
Yeah, which means that if you want to grow it in the lab, you have to grow a plant. Yeah, so that makes life a bit difficult sometimes. But it grows on a lot of agricultural plants. So we're looking at cereals like wheat, oat, barley, things like that. But there are other strains that can grow on things like coffee and other agricultural plants as well. And in Australia, we also have a problem with it's sort of come about that now there are strains in Australia that affect eucalyptus, so myrtle plants, so eucalyptus being one of them. And so you can parasitize those as well.
So it must be pretty important then to treat if it's a big part of a problem in the agricultural industry.
Yeah. Definitely.
There's a lot of money. Can I just ask, why is it an obligate, why is it such a parasite that without the plant it can't survive?
I guess it's just the way that it grows. It's just its survival mechanism is that
it's just the niche it fell into by accident.
Yeah. So in order to survive, it has to penetrate the cells of the plant leaves or the plant stem, whatever it's
It's similar, I guess, into a sense like viruses are even more specific, where they don't even, they might be for one organism, but they it's like one cell type even sometimes in that organism.
So
it's basically outsourcing a particular function that it can't perform itself.
Yeah.
And that's why it's obligate.
Yeah.
Okay. So it's a huge cost, obviously, to the agricultural economy. Now, how does so what are you doing in particular?
Well, how is that usually treated? Maybe we can go there first. How are we dealing with rust now?
So there's a couple of ways. So they can treat them with fungicides.
Mm-hmm.
So that costs the weed industry about, I don't know, $140 million a year to treat with fungicide, but they also can breed strains of the plant itself that are resistant to the rust. So what they do is they survey the rust that's in the environment at the time, and then they look at which plant strains are going to have some resistance to that rust. And so if they see that the rust is changing, then they're always kind of testing it to make sure that what they're planting in the field is going to be a strain that is resistant.
I guess there's a few problems with that though, because then you've got to try and predict what the strains are going to be, and then I'm guessing plant breeding like that is incredibly slow unless it's genetically modified, in which it's particularly controversial and expensive.
No, so they crossbreed the strains and then look for ones that are resistant.
So I guess that presents a need to kind of come up with better alternatives.
So it's okay when everything's going to plan, but then occasionally there'll be a new strain of rust that will somehow develop, and it'll kind of knock everyone for a six, and then they'll have to, they'll lose their crops for that year. And so, and then they go back to the drawing board. It's kind of like the vaccines for the flu, isn't it? Yeah. They've always got to be one step ahead of the flu, and they've got to know what strain's going around the world. And when it comes to Sydney, they give us a particular vaccine, and the flu might evolve into a different form, and then nobody's covered by that vaccine anymore.
Right. And so, what are you doing with your research in that context?
So, what's kind of interesting is that there are strains of fungi that are actually parasites of the rust parasite. So we call them hyperparasites because they're parasites of parasites. And so what they
just
do...
turtles all the way down. Right?
Yeah. Exactly. Yeah. So sometimes these hyperparasites just start to grow on a leaf that has rust on it, and we see that the rust dies. And so my collaborators were interested in how these hyperparasites actually reduce the growth or kill these rust parasites. And so that kind of is where it came full circle for me, is that I was interested in these small molecules. And I thought, okay, well, what small molecules are being produced by these hyperparasites that might be involved in, killing the rust?
So then to grow the hyperparasites, you have to grow a plant to grow the rust to grow that.
That would be a real pain. No, luckily for us, they're not obligate parasites. If they were, we wouldn't. So we just grow them on agar plates, and we're good to go.
Oh, that's good.
Sorry, I was just going to ask this. We'll come back to that point. Who found out that these fungi actually affect the rust? Was it you and your collaborators, or someone earlier?
No, I think that they've been discovered, quite a number of years ago. I don't know when they were first discovered.
It was probably farmers.
Yeah,
pretty
maybe.
Cluey farmers. Yeah.
Yeah, it's just something that's floating around in the environment. Just is lucky that it can land there and start to eat the rust.
Right. And how effective is this fungi at killing the rust?
It's very efficient, so it can kill the rust, but in order to be able to take the next step and say, let's use it as a biocontrol, it's not that, I don't think it's that efficient at going from one rust pustule to the next. So rust kind of grows as a little pustule, so it's like a little, pile of rust, a little rust spot on the
So it can't jump from one of those little pustules to a different pustule.
Yeah, so what you have to do is be able to spread the the hyperparasite evenly on the plant, or somehow, get the plant and the hyperparasite to make some kind of a relationship, symbiotic relationship, so that it kind of is nice and covering the plant.
So I guess that's where working out what the chemical is that's responsible comes in, because then you can develop that chemical, possibly synthetically produce it and distribute it as a pesticide.
Yeah,
is that right?
pesticide, but I guess we
It's herbicide for plant, and is it a pesticide for fungus?
Yeah,
okay.
fungicide.
You know lingo, don't you?
Oh, yeah. I actually listened to a great podcast called Talking Biotech with Dr. Kevin Falter. I think he's from the University of Florida. And he does, because he's a plant biologist, I think he works with strawberries, but he does heaps of great stuff about crops and plant science. And
they've looked at using mycoparasites, so parasites that kill fungi. They've looked at using those as biocontrol in the strawberry industry. So you can kind of inoculate your strawberries with this mycoparasite, and it kills off all of the
That's
fungi.
One thing that comes to mind is, do you think why that's valuable to go and characterize the compound? Would it be an issue besides going from one nodule to another? Would it be a biohazard to just let these fungi out in the wild to treat the rust? Is it going to be like one of those? What's the
cane toad?
The cane toad. Yeah, that's the other thing where I think in Australia we're a bit hesitant of the biocontrol. So
But if this is a naturally produced fungi anyway, then
it's a
already out
natural
there, isn't it?
fungi in the environment. But I guess it's one step from, if you have to spray it onto a field, that's kind of a bit scary, I think.
So how far are you into this project? Have you managed to characterize the molecule? Do you guys know the mechanism of action? How far are you into this project?
So we're just in the baby stages. So we've sequenced the genome of the strains, and we've isolated a couple of compounds. We're still in the process of confirming the structure, so we're not quite there yet. And our next stage is to look at the mechanism of action, and particularly because we need to know, okay, is it going to have an effect on human cells? Is it going to have an effect on plant cells? So if we want to use it as any kind of a agricultural compound or whatever, we need to know how it's going to affect other organisms.
So you said you isolated the compound, and that compound you've demonstrated actually kills the rust?
Yep.
Oh, okay.
And I guess the one further really exciting possibility is if you find out the mechanism of action, there's some potential there to either exploit the same mechanism or similar mechanisms and treat things like Hamid's ringworm.
Yeah, so once you have, what the target is, you can definitely exploit it. And I guess the thing is that a lot of the antibacterial drugs that we take are based on natural products. So these are, already being produced by a fungus, and then we take it and then we use it as a drug. But often those compounds are synthetically modified. So penicillin comes from a fungus, but then it's been synthetically modified to make it a better drug for humans. Similar
with aspirin, I think, isn't it? It comes from tree bark, but then it's been actually modified, because if you just eat the tree bark, you get really sick.
So I guess that's another stage as well. You can look at how can we chemically modify it to make it more suitable as a fungicide or an antifungal drug.
Are you taking master students and PhD students at the moment?
I definitely am. Come and see me if you're interested in this kind of stuff.
And sorry, go ahead.
Yeah, we're getting my current master student is getting ready to finish up, so looking for some fresh blood.
Awesome. So what type of projects do you have available? Do you have any specific ones in mind, or do you develop that with a student, or?
I guess sometimes it's developed with the student, but I guess, what we just talked about, looking at mechanism of action. But I'm also interested in whether these fungi produce other compounds that can be, so from our genetic analysis, we know that they can produce a lot more compounds. And what they do, we don't know. So, we've only just really looked for antifungals, but there might be antibacterials in there might be antivirals, there could be a whole range of compounds with different activities. So
the potential is big. Getting on
yeah.
that.
Yeah.
Cool. Thank you so much for your time, Michelle. We really enjoyed the conversation.
It's been a great conversation.
So
No worries. Thanks for
hit
coming.
Michelle up. You can find the details. If you can't find it, don't do research. Seriously. Bye.
Thanks a lot, Michelle.